Today I'm going to talk about a review titled Physiology and Diseases of Tissue-Resident Macrophages. Macrophages are a type of cell in our bodies that sense the environment, and they respond in a way that helps heal our bodies. Macrophages are essential in human viability and development. Macrophages have a range of receptors that can sense pH, temperature, osmolarity, hypoxia, which is the lack of oxygen, pressure, metabolites such as fatty acids, and extracellular matrix components. They can sense molecules that signal for apoptosis, damage, or unfit cells, and pathogen-associated motifs. The responses of these cells to certain stimuli include disposal of cellular waste, regulation of inflammation, secretion of proteases, and remodeling of the extracellular matrix components, as well as the production of growth factors. They can endocytose other cells or engulf them to help maintain a healthy environment. There are actually 3 waves of macrophage development. The first wave starts in the mesoderm during development of the embryo, and this is in the yolk sac, which actually sits outside of the embryo. These early macrophage progenitors express a gene called RUNX1, and these cells end up becoming primitive red blood cells and megakaryocytes in zebrafish and mice. So we don't know a lot about these cells in humans because we can't do experiments on embryos at this stage. Okay, so the next wave of macrophages come from erythromyeloid progenitors. These eventually become a variety of different cell types, as well as resident macrophages, which means they are macrophages that inhabit specific tissues. These also come from the yolk sac, at least in mice. These cells colonize the fetal liver, and then they form premacs, which are pre-macrophages that colonize the entire embryo and differentiate into their different cell types. And this happens during organogenesis. But the progenitors, the erythromyeloid progenitors, disappear during fetal life. The third wave of macrophages originate from hematopoietic stem cells. These are well-known stem cells that live in our bone marrow and can regenerate themselves. They give rise to a lot of different blood cell types, including macrophages. But these macrophages are different. They arise and inhabit different tissues based on the need at the time. Differentiated macrophages have different chromatin landscapes, which means that their DNA is modeled differently to allow for differences in transcriptions. And this gives rise to different functional characteristics of differentiated macrophages. There are lineage determining factors that will influence the way the macrophage differentiates. There are 2 hypothesized models that explain how macrophages differentiate. There's the instructive model, where tissue-specific growth factors reach Pu.1 expressing macrophages, where the chromatin landscape is primed to be accessible by lineage determining factors. So these macrophages are ready to be differentiated, and they just need the signal, which is given at the tissue that it will survive in. There's another model that the authors actually prefer, which is that these premacs, these are ones that originate from erythromyeloid cells. These cells are expressing different lineage determining factors, and they are circulating the body. And when they encounter a tissue, their lineage determining factor is a prerequisite for their residence in that tissue. And that is kind of what filters out the different macrophages and allows a specific macrophage to stay in a particular tissue. The bone marrow derived macrophages can replenish or replace the resident macrophages, but it is not something that always occurs, and the erythromyeloid cell lines persist in our bodies during our whole lifetime. But they can be assisted by the macrophages that originate in our bone marrow from the hematopoietic stem cells. The paper also outlines what happens when there are problems with particular macrophages. When there's absence of certain macrophages in various tissues, including adipocytes, which are fat cells, cells involved in bone growth and remodeling, disease can be a result. Macrophages are also involved in the clearance of fatty deposits in the lungs. It's involved in dealing with inflammation in the brain, as well as blood cell health. As a biochemist, it was really cool to learn more about cell biology and macrophages, and I hope to continually learn more over time. I want to have a better understanding of biology and physics and fields that overlap with biochemistry. I hope I didn't totally lose you in this video, and I hope you caught a glimpse of how complex and interesting macrophages are in human biology. I hope you had a great 4th of July, and see you in the next video.